12 has been neurons and how they work and what they are and blah, blah, blah. So this is one of our models of a multipolar neuron. Okay, multi because many dendritic surfaces, all right, and then it has one axon. Now, this axon connects in theory here, okay, and then continues on. All right, so as you go through, these little rubbery parts, these are axons terminating. So this part here, the hard part, the shell part, that is your dendritic membranes. Okay, so in these dendritic membranes, that's where you're looking to find your ligand-gated, chemically gated neurotransmitter receptors. Okay. Any of this membrane up here, again, that's you have resting membrane potential happening, so there could be some of these leaky channels and pumps. Okay. This is known as the cell body. This is the nucleolus, and so when you look inside, this is your intracellular fluid. Okay, your cytoplasm. Remember that the proteins in here, you would call them the neurofilaments, the neuroprotein. You know, you can put everything neuro, right? All right, these little blue things that are supposed to represent some message RNA, tRNA, sarcoplasm, rough ER, all right? These are some of your Nissl bodies that are little machines, little factories to potentially create the proteins, and they might have to slowly move because they might be the proteins that are being created to be put on the membranes or voltage-gated membrane channels, okay? So we don't necessarily want proteins always made up here by the nucleus, because remember, some of our neurons are going to extend a long way, so we want to make them closer to maybe their target location, okay? This area where we start to see the start of this long projection, this would be your axon hillock. Now, once you get into the axon, of course, you can start calling the cytoplasm the axoplasm. You can start to call the membrane here the axolema because it's the membrane of the neuron related to the axon. So they can start to have vocabulary specific to the axon. Okay. Now, this is a myelinated axon. So these are supporting cells. Now, these neuroglial cells are like a pancake wrapped around a specific location. So these neuroglial cells represented here should be Schwann cells. And if these are Schwann cells, then this neuron, its axon, is going to be in the periphery. The cell body might be in the spinal cord, but most of its axon, if there's Schwann cells around it, is going to be part of some type of cranial or one of your spinal nerves. So this might be the axon heading towards your forearm to control one of your flexor or extensor muscles of your arm. Okay, and it might be part of the radial nerve or the ulnar nerve and, you know, brachial nerve, and come out maybe around C6 or something, okay? Now, remember that I do not want neurons to touch each other. And I don't even want their supporting cells and that little space, the node of Ravagnier, I don't want any of that electrical material to potentially touch or get too close to other neurons. Because if I'm going to control skeletal muscles, I want only the 5 or 6 maybe skeletal muscle cells from that one neuron to contract and not any surrounding, right? So remember that just like in our skeletal muscle organs, when we look at our nerves, when we look at your cranial nerves, when you look at your brachial nerve, your ulnar nerve, your sciatic nerve, it is composed of a bunch of myelinated and unmyelinated axons. And to keep those axons from potentially having electrical information jump inappropriately to other neurons, individually they are wrapped in connective tissue. So this gray area here represents the connective tissue around a single neuron and its supporting neuroglial cells. So in a muscle, the connective tissue around one skeletal muscle fiber we called endomyosin. Okay, this connective tissue is still endo, but it's around a neuron. So you're going to call it endoneurium. Sounds like neuro, okay? Now, again, if I was to take any of my nerves and do a transverse cut, I'm going to see that many nerves are grouped together. And some of that's because, okay, I have all these skeletal muscle fibers that need to go towards this muscle. These need to go towards this muscle. I have all these sensory informations coming from my pinky versus my thumb. So I organize them into groups, and then I wrap them in connective tissue. Those groups of axons, and again, they might represent groups of some axons all going to, all going away, or mixed. Those are going to be fascicles. And fascicles, just like in a muscle, are wrapped in connective tissue that is perineurium. And then the entire nerve, which is normally going to run with the blood vessel and the vein, the artery and the vein, is going to be wrapped in more connective tissue. That is going to be epineurium. Okay, so anything listed in your lecture notes in that lab manual related to landmarks on a neuron, and then this is represented for the endoneurium, that would be one station on your lab exam. Okay, now, just because we have them, I don't usually test on them, all right? On here, this is your eyeball, okay? And remember, I told you you have rods and cone cells, so that's represented here, okay? And then you have these neurons where they have a projection on one side of the cell body and then the projections on the other side. What kind of neurons were these? Do you remember? These are bipolar neurons. And this is one of the only locations we have them, is in our eyeball. So here's a picture of a bipolar neuron. Then you get to another type of neuron here, where again, here's the dendritic part, here's the cell body, and then here's the axon part. And the axon part is going to run and become your optic nerve. And so within your retina, you have a synapse between the rod or cone cell and the bipolar neuron. And then you have a second synapse between the bipolar neuron and what's known as the ganglia cell. So there are two synapses in your eyeballs before information even leaves your eyeball to head towards your occipital lobe. Okay. This model is not on the test. I'm just showing it because that was a multipolar neuron. This is what a bipolar neuron looks like. And it's one of the only locations we see it. Okay, let's move to the spinal cord next. Okay, now there will be one station on your lab exam with the model. There will be one station with the picture. So there are two stations on your lab exam. Okay. Okay, so let's go through the spinal reflex because that's what it kind of shows you here. It shows you that for your spine, you are going to have afferent sensory neurons connected to temperature receptors, pain receptors, itch receptors, tactile corpuscles, laminated corpuscles, all right? And they are going to bring information in, and that information is going to be, remember the little leaf, that unipolar neuron? That little, like, it has this long extension. It looks like a little leaf-furred cell body, and then it has its axon extension, all right? That little leaf is going to be for all of those afferent neurons on this spinal nerve. They are all going to gaggle together right here. And if this is the spinal nerve and it has two roots, this root is going towards the posterior or the back of the body. And in animals, what do we call posterior? Dorsal. So this little gaggle is known as your dorsal root ganglia. It is going to be full of the little cell bodies of all those unipolar sensory afferent neurons bringing information from this nerve into the spinal cord. And as the picture shows, your sensory information comes in and it's going to synapse on the posterior of your spinal cord in this gray middle piece. Okay, this area is known as your gray matter. Gray matter is where we normally don't expect a lot of fat to be present, so there's not a lot of myelination here. So this is going to be the area where we have a lot of terminating axons, and at the termination of the axon, we don't usually continue to have myelination over them. And then we have a lot of cell bodies. And again, if you look back at these multipotent neurons, are there a lot of myelinating parts on the cell body in the dendritic membrane? No. So in your gray part of your spinal cord, you are going to have a lot of terminating axons and a lot of cell bodies. Okay. If you divide the, again, spinal cord in half, you're going to have a dorsal and an anterior. One of the ways you can tell the dorsal is look for, if it's present, the dorsal root ganglia on your spinal nerve as it comes in. The second way to try to find what's dorsal versus anterior, where you have cut on midline, the posterior material for the white matter, it's a sulcus. A sulcus means everything's touching, but there's an indentation. Okay, they're pushing in and they're forming. So if I put a butter knife in there, can I squeeze a butter knife in between these two things? Yes, that's a sulcus. If I look at the anterior, I am going to have a more pronounced space between the two items. So sulcus, it's like a butter knife, everything's touching and making a little groove. Fissure is a deep groove. So this is going to be my posterior or my dorsal. It's on midline, dorsal, median, sulcus, or my posterior median sulcus. This is my anterior median fissure, or my not proximal. I'm like, what is it? It's not anterior, it's ventral. Ventral, yes, thank you. See, brain fart. Ventral median fissure. Okay. Okay, third way to try to tell anterior, posterior, all right? You look at the gray matter. The gray matter is going to have points that come off. What do we call points if they come off a head? Horns. So the points are going to be horns. You are going to have on each side, the right and the left side, you should have a horn that points towards the posterior. So that's your posterior or your dorsal horn. You're going to have a little bit of a rounded but a pointy part towards the front. That's going to be your anterior horn. And usually there's a horn that goes out towards the lateral part. So that's going to be your lateral horn. When you look at the gray matter, the anterior horn is never usually going to reach near the surface of the spinal cord. So your anterior horn is going to be more rounded, and it's going to have usually some white matter between it and the margin, the edge. On the posterior side, it's going to get really close, and sometimes it will actually be at the margin. So when you draw it out, if you look, think of it as a butterfly. These are usually like the little rounded wings, the inferior wings, and these are like the big pointy, top wings. Okay, so 3 ways to be able to tell anterior, posterior. If you have the spinal roots, you look for the dorsal root ganglia on the posterior root or the dorsal root, and then this is the anterior root. You look for the anterior median fissure versus the posterior median sulcus. You look at the gray matter versus the white matter. the gray matter should have, again, close to the margin, the posterior horns. Okay. Now, think back to our spinal reflexes. I touch something that's really hot or really cold. My hot and cold temperature pain receptors are going to bring in the information. They are going to synapse at the posterior horn on a few interneurons. Remember, one of those interneurons is going to start to go towards my brain to tell me, ow, right? One of the other ones, because if it's my foot, one of the other ones needs to cross over to the other side of the body to tell the other side of my body, like, get ready, you're about to have to take the weight, right? And then one of my other interneurons is going to come around to the anterior side, same side, and send out a motor, skeletal motor neuron to contract to pull my hand away from that stimulus. Okay, so this gray matter represents where those interneurons exist that help them connect the posterior to the anterior on the same side, the posterior to the cross extensor to get to the anterior on the other side. All right. Now, my neuron going to the brain, it is going to have an axon that's probably myelinated. That myelinated axon is not going to stay in the gray matter because if it's myelinated, it's going to be white matter. The white matter part of my spinal cord, if you think of axons as like a bunch of spaghetti noodles, they are all gathered together and they are all running like spaghetti noodles in a jar, north and south, and they're wrapped in oligodendrocytes. Okay, so if I was to try to grab a bunch of these spaghetti noodles and pull, they are all arranged as a column. They're all running north and south. So the area between my dorsal horns over here, right by that sulcus, this is known as my dorsal columns or my posterior columns. Most of these neurons are carrying sensory information to your brain. Same thing in the anterior. There's a bunch of, again, myelinated axons. Most of them are carrying information from your brain. And then you have some other pathways in the lateral columns. Okay, in general, if I cut the spinal cord in dorsal, ventral, or anterior, posterior, the back of your spinal cord is sensory. So if you get an epidural, do they try to go to the front of your spinal cord or the back of the spinal cord? The back. They're trying to numb your columns and your area from that level and below from receiving sensory information. So they are just trying to stick that needle in and shoot out medicine to disperse towards the dorsal part, and it cuts off feeling. Technically, could you still wiggle your toes? Yes. Okay, you could still technically move because anterior is going to be where motor and autonomic and everything unconscious and conscious movement is going to flow. Okay, so I always like to have, especially in lecture, I like to do a question of, if I was to completely give you a viral infection and kill the back, the posterior part of your spine, are you going to be able to still walk or not? Yes, you can walk because the anterior is where motor is. Are you going to be able to feel any temperature, or pressure, or pain anymore? No. Okay. If I give you a viral infection and I take out most of the anterior part of your spinal cord, are you going to be able to walk? No. But will you still be able to feel what's happening to your legs and your arms? Yes. Okay. All right. So as you look at pathways that are going to the brain, the afferent going in and to the brain are going to tend to be on the back, the posterior columns, some of the lateral but posterior areas. Information coming down from your motor cortex, from some of your autonomic systems, those are going to tend to be on the anterior part of your spinal cord. Okay, now It gets a little more complicated when you go from midline, the right side and the left side. There are some pieces of information that when you synapse, the inner neuron crosses over. So I carry some sensory information opposite side of my body up to my brain. Other sensory information I carry same side of my body, and then it crosses over at the thalamus. So if you were to lose half of your spinal cord, like the right half, you would still have some sensory information on both sides of your body. You would have pain and temperature on one side, and you would have, I forget exactly, pressure and touch on the other. You would lose motor on that side from the injury point down. Okay? Because you would lose all of these motor neurons that go out and control the skeletal muscle. But sensory, you would lose, again, if you lose the right side, you're going to lose some sensory on your left side and some on your right side, because some sensory information crosses over at the spinal cord, some waits to cross over at your brain. Okay? Now, in general, if I'm right-handed, what part of my brain is controlling my right hand? My left brain, yeah. Okay? But that crosses over at the brain and the thalamus. So if I have an injury at the spinal cord, I'm going to lose everything from the right injury to my right side for motor. Okay? It is really cool if you get into spinal cord, because we have mapped out, in addition, if I was to only kill the parts of the column that's closest to the posterior median sulcus, I can tell you exactly, like it's mapped out exactly at that level, what sensory information you've lost and at what part of the body. Okay, so there is a lot of mapping, and everything's mapped for the body, and then it comes into your brain. And if you've ever seen again, for the brain, the primary motor cortex and the primary sensory cortex, they're all mapped. So if you if you see people with those little caps and they send electrical impulses, they can make things move, and they can make you feel things by sending little shocks to your brain. They've mapped it all out. Okay, did I cover everything on that spinal cord sheet? For the most part. Central canal, so you're going to have cerebral spinal fluid in there, all right? And then remember that all of this is wrapped in meninges. And it's inside your vertebra and inside that space. So bone does not technically touch oligodendrocytes and neurons and axons, okay? So you're going to have astrocytes around the blood vessels. You're going to have meninges, and the meninges are going to be the closest to the thing is the pia. Then you have the arachnoid with the subarachnoid space, and then you have that dura, which is the outermost. Okay. All right, we'll go through more of that in lecture. All right, good with this? Okay. All right, now we can do brains. Okay, there are 4 major areas of your brain. So if you take zoology, you start looking at worms and you start looking at mollusks, and then you build up to bigger entities. Okay, the most archaic part of our brain is our brain stem. Okay, our brain stem is going to have this area on here. It's like labeled D. That's known as your midbrain. And we don't know a lot about the brain stem and the midbrain because, well, in simple organisms, when you poke it, you get like, they wiggle. I mean, like, you know, there's not a lot of like, they can't tell you what they're doing, right? And as you get to higher multi-complex organisms, this place is very protected. So it's hard to like poke holes in there. We think the midbrain is tied to some reflexes. Like if I hear an ambulance, what am I generally going to do? Look towards the sound, right? So like think about deer. If it hears a hunter, what does it do? It picks its head up and it looks towards the sound. That's like a reflex to try to save yourself, right? From a predator. Okay, so we think that some of that comes from the midbrain. All right, the next part is labeled E here, and it looks like a little almond. That is your pons. Okay. there are going to be some connections between this part and your cerebellum, right? And there's some connections and some stuff here that might influence your breathing, some higher level breathing. Right. The next part is F. That is your medulla oblongata. And if you've seen the Bubba, what is it? The water boy. This is not where anger resides. Okay. This is the part of your brain that does a lot of that autonomic function. So you need to raise heart rate. There's a cardio center here. You need to change your breathing rate. It does that. So some of your reflexes of baroreceptors, chemical signaling for how your heart stretch, your pressure, and your chemical makeup of your blood, it's all going here. And you will exit out here a lot of parasympathetic and sympathetic influence. Okay. So coming out of here is going to be things to like make blood pressure go up and down, make breathing rate go up and down. A lot of those things that when you sleep, it's keeping things going. Okay, so that is the brain stem, those three parts. Okay, now, next part of the brain, okay, is like this. If this is like a little bird, it's this part. And again, you have to think three-dimensionally. There's two eggs and then this center material. Okay, the two eggs are the thalamus. You have two thalamus, and then the central area that, as you can see in this guy, is then going to connect to this little round thing. This is your pituitary gland. This is the diencephalon. This area would be your hypothalamus. Here's your pituitary gland. And then the little thing that looks brown, think of this as the butt of the egg, and the egg goes that way. So if you're thinking about you pull an egg out, you're seeing the top or the bottom of the egg, and it goes that way. It's sitting on its side. That's your thalamus. Okay, so your diencephalon is going to have two thalamus, hypothalamus, the mammillary body, the pineal gland, and your pituitary gland is all associated with that. Okay, so we learned from last chapter of 18, hypothalamus and pituitary gland do a lot of control, right? There's hormonal, and then the hypothalamus also has some, from chapter 1, set point, homeostasis. So this is a big, if this is your CPU for basically keeping the temperature, the water, the things going. Okay? All right. Now, next part of the brain is on the back. All right? This part's the second largest part. This is your cerebellum. Okay? Now, your cerebellum, you start to have lobes. So there's like an anterior lobe, a posterior lobe. They're on midline. There's some fibers that run and connect the right and left cerebellar lobes. That's known as your vermis. Okay? Now, your cerebellum is going to be the opposite of your spinal cord. So remember, your spinal cord had gray matter on the inside, white matter on the outside. Your cerebellum is going to have the gray matter on the outside, white matter on the inside. Because think about it. Information is coming into and out of the cerebellum. That's going to travel on myelinated axons. The parts that are unmyelinated are going to be the receiving or the initiating cell bodies and material. Now, if you look at the white matter in the cerebellum, it looks like a bunch of branches of a tree. Does anybody know the word for tree in French? We have a day for it sometimes, about tree day, arbor. So this is known as the arbor vitae, because it looks like tree branches, arbor, and vitae for life. Okay, cerebellum is going to tie with motor. So I am going to initiate that I want to grab something. The cerebellum is going to keep me somewhat from shaking and overshooting and missing. So the cerebellum gets some information from my ears about making sure that as I reach for this, I also keep my balance. And then I just smooth out the muscle contraction so everything isn't jumping. Okay? So when people start having cerebellar issues, they walk really, really, really funny. And it's because the cerebellum can't balance out the smooth transition from the right side to the left side and can't maybe do their balance and their weight distribution as they are moving. And so people with cerebellar issues, so what happens when old people get old? They fall a lot, right? And we scan their heads, and sometimes with their falls, we find, oh, you got cancer from the neuroglial cells in your cerebellum. Or you had cancer somewhere else, and it migrated, and now it's taken up residence in your cerebellum. Okay? All right. In humans, the biggest part of our brain is the cerebrum. Cerebrum. Okay. Your cerebrum are all these lobes. The big landmark is, here is the white matter that is going to connect the left and the right hemispheres. That is known as your corpus callosum. Everything above and over that corpus callosum is part of your cerebrum. Okay. Your cerebrum looks like it's a bunch of cheese squished together, right? So the indentations are sulci, sulci. The cheese itself is going to be called the gyri, or the gyrus. Right hemisphere, left hemisphere. There is a big groove. That big groove is your longitudinal fissure. Now, there is a pretty significant sulcus that runs right about where you would think a coronal or a frontal cut should happen. And there's a gyrus in front of it and a gyrus right to the back of it. That is going to be known as your central sulcus because it's not as big a groove. That is the landmark that's going to make your frontal lobe versus your parietal lobe. So your frontal lobe is going to be from the central sulcus towards the front. It's underneath the frontal bones. Your parietal lobe is going to be from that central sulcus towards the top, towards the back. Now, there's not as big a division, but there was some other sulcus, lambdoidal sulcus, that is coming into play for the occipital. Okay. All right. Some books call this a fissure, some call it a sulcus. So I think you can get away with either. What is your lab manual calling it?
Lateral sulcus.
The lateral sulcus. Again, sometimes the books will say it's a fissure because it is a little pronounced between the temporal and this. Okay, so the temporal lobe is going to be again right at the behind the temple bone, and then the occipital lobe will be again underneath the occipital bone. Okay, coming back to my frontal lobe. That gyrus in front of the central sulcus, it's in front, so it's the pre-central gyrus. This gyrus behind the central sulcus is known as the post-central gyrus. This pre-central gyrus, just like with the cerebellum, if I'm scratching on this, I am going to be touching gray matter. So the white matter's to the inside, and the outer material closest to the surface is going to be gray matter. That outer third of gray matter is known as your cortex. So this outer cortex of this precentral gyrus has been mapped, and this is where I control most of my skeletal voluntary muscle movements. So you want to wiggle your thumb, you want to stick out your tongue, you want to move your lips. This is that control, right hemisphere, left side of the body, left hemisphere, right side of the body. There are 3 gyri that sit kind of like my fingers are running perpendicular to that central gyrus. So these, we think, are some of the gyri that are going to plan if she wants to jump up, this is what we do. If she wants to move her head and start dancing, this is what she's going to do. So they are making the contingencies for how you're going to move next. So they are like before you're thinking about what you're going to move, they're making the contingencies for how you would move based upon what you're doing.
Wait, is that what the precentral do?
This is, again, no, precentral is the primary motor cortex. So this is causing, right now, you blinked your eyes, these made you blink. These make you move your lips. These make you move your lips. This in front is contingencies for what are you going to do next based upon where you are right now. Okay, so this is like the motor planning before you actually motor plan and do something. Okay. Then the very front, that's what you do in psychology class, right? Who are my psychology people? You talk about higher mental brain processing, personality, the artsy side versus your logical, analytical thinking. So do I care about that in physiology? No. So who do I make you take classes to learn about that crap? Psychology. Okay. But if you start having, again, when they drill into people's brains right here, I am right-handed, so I'm left sphere dominant. So if they were to probably put a bullet right here in my right hemisphere and my very frontal lobe, I might tolerate that if it got stuck there. Not saying let's do an experiment, but but if you hit me here, I would probably potentially have a big, if I survive, I might have a big traumatic personality change. Because how many of these neurons die, and that's my dominant hemisphere, and that's where dominant my personality, my logical thinking self is. Okay. All right. For the parietal lobe, again, that postcentral gyrus. So if this is where my lips are controlled for movement, this is where my sensory information from my lips is coming. So this is my primary sensory cortex, getting information from touch, pressure, information, and they're usually going to be mapped. So if over here is my pinky information, well, this is where my pinky is controlled. All right, and so that information, if something's going on sensory, it can very quickly jump and make motor, or if something's going on motor, I can very quickly sense it. Okay, so in yoga, every now and then when you're talking about meditation, every now and then in meditations, you do body scans. There's a certain way they try to teach you sometimes to do the body scans because they're trying to get you to start in here in the longitudinal fissure where these sulci and gyri start and work your way out, versus there's other ways where you start here and you work your way in. So you start like right side and you go and then you hit the left side. So there's a body mapping way in meditation where you're trying to like take a tour of the brain in these areas. Okay. The back or the occipital lobe, we know that that's where vision is. Okay, so if somebody gets hit in the back of the head, they sometimes have like that blacked out, they can't see for a little while, they have a hard time focusing. That's your primary visual cortex. Okay, now in the temporal lobe, we know that that is going to be, we have some taste, we have hearing, so there's some areas that are known as our primary taste, our gustatory cortex, and then we have areas for our auditory cortex. So when you think about someone getting their cochlear implant, a lot of times that's right here in that temporal bone because they're trying to get to that auditory cortex. Okay, and then sometimes you have to like pull it up, and again, three-dimensionally, there's neurons and material in that sulcus sitting there. All right, now, when you pull this off, back when I did, you can kind of see it has this letter A right on top of the corpus callosum. So back when I first did this, they just kind of said this was part of the frontal lobe, this was part of the parietal, and then the part down here would be part of the temporal, and then this was part of the occipital. Now, some books are saying this one gyrus that sits right and wraps around the corpus callosum is a different lobe. So some books are going to call it the limbic system. Some are going to call it the limbic lobe. Now, in this system, you'll have your hippocampus. Okay, so we know that somewhat sleep and memory processing are in here. The other thing we know from serial killers, because serial killers have messed up arousal and happy things. They associate sometimes, let's kill somebody, and that makes them excited and aroused. And that would be an inappropriate response for normal people, right? So we know that somehow your memory and your emotions get tied together in the limbic system. So when it is not working correctly and tying potentially sexual arousal and happiness to society norms, bad things, that, you know, that's part of the reason people have messed up limbic systems. Okay, so the big debate, your generation is all about video games. You've had video games and first-person shooter games your whole life. Supposedly, you have a limbic system that's been desensitized to shooting people. Okay, because you've been trained that it's okay in this video game concept to shoot people and kill them and see blood. All right, that a little bit before my generation, but Generation X, Generation whatever, the baby boomers, they didn't have video games, so they supposedly have a different limbic system tie-in to that. And you might get excited in shooting because you've been conditioned that it's exciting to kill in a video game. So that was one of those big things about video games, all right? So that's the limbic system. Okay. As you go through the rest of the chapter, you're going to see on the dominant hemisphere, and it kind of has some different little circles on this left side. We know that there is some type of area where a bunch of the lobes come together, known as the general interpretive area. That is where we think, for most people, your perception of reality is occurring. It's sometimes also known as Wernicke's area. We also know that somewhere in the frontal lobe up here on your dominant hemisphere, there's an area known as Broca's area, and that is where you have your speech center. So if you were to get a frontal lobe injury and you lose Broca's area, you would lose the ability to speak. Okay. All right. So again, how did we figure this out? A lot of it was done by the psychologist. A lot of it was done when they cut the corpus callosum and they talked to the right brain or only the left brain, and they started to gain, well, when I talk to the left brain, I can have them talk to me. When I talk to the right brain, they can't speak, but they can recognize characters and draw, all right, or write things. So that's part of those epileptic, those studies with psychologists came in handy. Okay? Brains for the most part, all right? Again, cranial nerves are on here. So you can see the cranial nerves, your 12 pairs of cranial nerves. We're going to go through that next week. They come off of the cranial areas, and you have to know them by name, by number, and you need to know what they test. It's really fun. Ooh, 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 because the first three all start with the letter O. So what do you come up with a sentence or a mnemonic device that has 3 O's? O, 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 ooh, ooh, ooh. Okay, so there's some fun little things out there to try to help you learn them. Okay, so we'll get to that. All right, now, your meninges around your spinal cord wraps around your brain. Same 3 layers, okay? In your subarachnoid space, remember there's cerebral spinal fluid, right? Inside the brain, you have cerebral spinal fluid sacs. These are going to be your ventricles, okay? Now, the right and the left lateral ventricles, we don't give them a number because if I tell you this is number 1, a subconscious bias is going to say 1 is better than 2, and is 1 really better than 2? No. So these are just the lateral ventricles. They should be the core, the areas that show it kind of pink. Those are where those epidermal cells are that are helping to create the cerebral spinal fluid. Okay. Now, these ventricles are connected to your central canal, and they are connected to your subarachnoid space. So the fluid that is completely inside and outside the brain. So the brain is completely like floating in fluid. And that helps when I shake my head. Does my brain bump into my bones and get damaged? No, because that water kind of insulates it and shock protects it. Okay, again, shaker babies, babies with little brains, you shake them too hard, they will have damage. All right, concussions, you hit your head hard enough and it will get damaged. But for the most part, if we're shaking our head in the club, like, you know, what was that movie? You know, we're not causing brain damage because our brains are suspended in cerebral spinal fluid. Okay, so you have the lateral ventricles. They're associated with the cerebrum. Okay, they are connected, and then you get on midline and you're at the third ventricle. The third ventricle is associated with the diencephalon area. Okay, so that's the thalamus, the hypothalamus. All right, and then you're going to have a connection and you're going to get to the fourth ventricle, and the fourth ventricle, you can actually see it right here. It's that little bit of tubing. It's associated with the pons and the medulla in the brainstem. Okay. I don't remember exactly, but I know there's a cerebral aqueduct. And what does it say the cerebral aqueduct connects? Okay, so the connection between the third and the fourth ventricle is the cerebral aqueduct, and then it's the interventricular foramen, right? Those should be the connections then that connect the lateral ventricles to the third ventricle. Am I right? Did I get that right? Okay. Now, where the fourth ventricle is going to eventually go in and become the central cavity, there's a little bit of a close space here that it's also going to bleed into the meninges. When someone has too much cerebral spinal fluid being made, so if I make 500 mils of cerebral spinal fluid, I should be taking away 500 mils of cerebral spinal fluid. But if I'm making too much cerebral spinal fluid, that fluid's going to have more pressure and it's going to push. Bone doesn't give, right? So it's going to push on my nervous system. Okay, so if I have to put a shunt and I have to let cerebral spinal fluid leak out, they're going to do it in that brain stem really close to where your spinal cord starts. So at the base of your skull, they put a little bit of a puncture, a little bit of shunt, and that fluid can slowly leak out and become part of your interstitial fluid. Okay, it's risky because if I have a puncture there permanently, can antibodies, I mean, things should be by pressure flowing out. But I do have potentially a puncture that if something was to happen, T cells and B cells could get inappropriately where they shouldn't. Okay, but sometimes you have to take that risk because you don't want to kill neurons. Okay, I've been talking for 51 minutes. Questions? Okay, so this will be one station. Definitely going to be probably 1 to 2 stations on landmarks and features, gross things. Cranial nerves will be a few stations. We'll get there next week. This will definitely be 1 station. Spinal cord, definitely 2 stations. So I have 1, 2, 3, 4, 5, 6. I have at least 6 out of 15 to 20 stations right here, right? Everybody cool? Any questions from you guys online before I'm going to stop the recording?